Hey guys, in this video I'm going to be focusing on an aspect of the coronavirus which has a pretty good connection with what we've been talking about in class, or what we were talking about in class before class ended. And what we're going to be talking about is the interaction between the spike protein, or the S protein, and how it resides in the membrane of the virus. All right. So just a little reminder: the spike protein is the protein that embeds itself in the membrane of the virus, and it is responsible for binding, for the specific binding of the virus to its cellular target. All right. So without this spike protein, there's no way for the virus to infect anything, because it can't bind to the host cell without that. All right. So the thing about this discussion is it involves two types of biological molecules that we've talked about. First of all, it deals with proteins. And the second thing is it deals with a type of lipid called a phospholipid, which we mentioned back earlier. So let's do a little review of those two things, and then we'll talk specifically about why is it that this spike protein wants to stay embedded? Why don't these proteins just float away from the bilayer? So first, a little bit of a review of what makes up both the cellular membranes of the host cells. This would be, for example, our cells, the ones that are going to get infected, but also the membranes that surround the virus. All right, and recall that these membranes are made of what are called phospholipids. All right, so a lipid is a triacylglyceride. All right, so in red right here, you have essentially a molecule of glycerol. And if this were to be a regular fat molecule, you would have three fatty acids, which are connected by ester bonds to that central glycerol. All right. So what makes a phospholipid different from a standard lipid is that one, instead of having three fatty acids, you have two fatty acids, and then that third glycerol hydroxyl group has a phosphate connected group to it, and this part of the molecule right here is what's called choline. All right, but the important thing to remember about this is that this part, with these long chain fatty acids right here, this part of the molecule right here is very hydrophobic. All right, it's made up of just carbon and hydrogen bonds, and what it means is they really don't like water. All right, there's nothing. There are no polar bonds in this part of the molecule, but this part of the molecule right here is a water-loving part of the molecule. It's very polar. You've got these phosphorus-oxygen bonds, which are very polar. It's also got this, although they don't show it, there would be a positive charge on this nitrogen right here. All right, so this part of the phospholipid really likes water. All right, and a lot of times, so even though this is the molecular structure of a typical phospholipid, a lot of times it's represented like this. All right, where this straight line right here would represent essentially a fully saturated fatty acid. This bent part right here, the reason for that bend is that there tends to be a cis double bond in that particular fatty acid. So that's what this part right here is. And then this head right here is what is used to represent this polar part of the phospholipid. That's the phosphatidylcholine part of the phospholipid. And just the important part to remember is this part right here, which corresponds to this part, loves water. It really likes to make strong intermolecular interactions with water. But this part of the molecule down here really does not like water. All right, and so what ends up happening is that if you were just to take a bunch of these molecules, enough of these, and throw them together, they would tend to want to arrange with each other. And so what this slide shows is 6 molecules of phospholipids that are arranged with each other. All right, so here is one right here. All right, so you can, this and this are the two fatty acid chains. All right, the glycerol molecule would part of the molecule is right here, and then this part right here would be that phosphatidylcholine. That's the polar head group of that phospholipid. All right, so notice that so you're going to have three of them side by side here. So here's one of them right here. Here's another one right here. Here's another one right here. But then directly opposite of those, you're going to have three of them on the other side. All right, arranged like this. And notice the reason that they do this, first of all of these long chain fatty acids, they're hydrophobic. London dispersion forces make it so that they like to be around each other. All right, they would much rather have interactions with each other than have interactions with water, because they're very non-polar, and water is a polar molecule. All right, so first of all, they like to stack next to each other, but if you think about the tips of these things, that's also a very hydrophobic part of the molecule. And so instead of that side right there wanting to be exposed from water, well, what if we had this other side line up? Well, that would put a hydrophobic part of this fatty acid right next to the hydrophobic part of this fatty acid, and that's a very stable relationship. All right, so the London dispersion forces both contribute to the reason that they want to stack up side by side, but it also is the reason that they want to arrange themselves in essentially two layers. All right, so this is what is eventually going to make up the cell membrane. All right, because this thing is going to make can eventually circle around. It can circle around, and it can essentially make a barrier between a water environment on the outside of a cell or a virus from the water environment on the inside of the cell. Remember, so if you take a look at this right here, all those fatty acid chains are sandwiched in the middle of this what they call a phospholipid bilayer. All right, the reason they call it a phospholipid bilayer is that there's two layers of the phospholipids. There's an outside layer here, then there's an inside layer. All right, so it's that polar head group that's in contact with the polar water molecules, but it's the non-polar fatty acid chains which are in the middle here. All right, so the one thing that you need to remember about this is that this region right here really does not like polar groups. All right, that's why it makes a great membrane. All right, you can separate one water environment from another water environment because the water that's inside here, it really doesn't want to go from this side to this side, because it would have to travel through this region right here, which is really not that friendly for water. All right, so what I'd like to focus on in this video specifically is the nature of how proteins get embedded in this phospholipid bilayer. All right, right now if you're to look at this diagram right here, it looks like this is a perfectly happy relationship here. This thing would not want to be disrupted, but we know that, for example, with the spike proteins on the coronavirus, that proteins often will embed themselves in this bilayer. All right, and how does that happen? All right, let's first go to a little review of the nature of proteins. So first of all, before describing this, I just want to point out, like, if you wanted a more in-depth review of proteins, you can rewatch the videos that were on the protein basics page. But I'm just going to go over a couple of slides that give a quicker review of that. So first of all, recall what a protein is. A protein is a molecule that is essentially a chain of amino acids linked to each other. All right, so amino acids are shown here in the data booklet. And remember, all amino acids have there's something that they all have in common, and there's a way that they're all different. All right, the thing that they all have in common is that you have this nitrogen, this amino group right here, bonded to this what's called the alpha carbon, and then you've got a carboxylic acid over here. And every single one of these amino acids, there's around 20 some of these amino acids, is going to have that same commonality. The thing that makes them different is the what's called the R group that they have. All right, notice that each of these has got a different group at this position right here. All right, and so when you have a protein, what's going to happen is that the amino side of one of these guys is going to form an amide bond with the carboxylic acid side of another, and it's just going to make a really long chain. And the only thing that makes the amino acids different is what that R group is. So this slide right here just shows a bunch of amino acids which have been linked to each other. Again, when they're linked to each other, you don't see a carboxylic acid in an amine. So this would be formerly the carboxylic acid side of one of the amino acids, and this would have been the NH2 or the amino of a neighboring amino acid. But when they form that amide bond between each other, they lose a molecule of water. But this is now what you call a peptide bond. All right, and essentially that's what that's the first way to understand a protein, is that it's just a chain of amino acids that are bound to each other. All right, and the sequence of this is what makes up what's called the primary structure. So again, in this slide right here, we can see the order, just the sequence of the amino acids linked to each other. But once you get a long enough chain of amino acids, these amino acids can start to take on another level of structure. And one of the primary ones is what's called an alpha helix. All right, so here is the chain of amino acids, and it's just kind of winding around like a corkscrew. But one thing that can happen is that the carbonyls of some of the of an amino acid here can hydrogen bond to the NH group of an amino acid that's several amino acids away from it. And you can see all these dotted bonds. Those are hydrogen bonds between neighboring, between close by amino acids, and that really acts to hold this structure together. Remember, hydrogen bonds are little bits of Velcro that create little areas of stickiness between atoms, and that holds it together in this overall corkscrew shape called an alpha helix. All right, there's other types of secondary structures. Another common type is a beta pleated sheet. But then once the protein gets long enough, even the secondary structures can start to interact with each other, and that's what creates what's called tertiary structure. And if you get, so tertiary structure is just the three-dimensional shape that you would get from a bunch of secondary structures interacting. For example, you might have an alpha helix right here and another alpha helix right here. And then, although I don't see any representations of beta pleated sheets, you could have some of those over here. All right, but within this would be all one chain. All right, but occasionally in proteins, you get multiple chains that interact with each other. So this would be like one chain shown in the dark green right here. This would be another chain right here. All right, and when you have multiple chains of amino acids interacting with each other like this, that's what's called the quaternary structure, and that's going to be fairly relevant to the spike protein, because the spike protein is actually made up of 3 different subunits. So if you were to look at the virus from a distance, a lot of times you'll see illustrations where they just represent the spike protein something like this. And again, this is another one of the common illustrations. These red areas right here, that's what the spike protein looks like. Notice that it's embedded into this gray area, which is the membrane of the virus. So this is a little bit of a closer up image of the spike protein. And remember, there's that quaternary structure that's possible, which is 3 separate chains that are separately folding around on themselves, but then interacting with other chains. All right, and you'll notice that there's 3 different colors here. There's a purple color, there's a light blue, and there's a yellow color. And what that indicates is that there's actually 3 separate chains of amino acids that are interacting. So this protein definitely has what's considered quaternary structure to it. right, we'll continue a little bit more in the next video.